I'm Franziska Trede. I'm a PhD student here at the German Primate Center.
My name is Niels and I'm a PhD student at the University of Göttingen and the DPZ.
My name is James Stranks and I'm a PhD student at the University of Göttingen and the DPZ.
In our paper that we recently published, we created a new panel of microsatellites that can be applied to all old world monkeys, including great apes. And this panel can be used to analyze the genetic variation in those primates and makes it more comparable across species.
Now, there's several reasons why it might be important to know the genetic relatedness between individuals. For instance, in breeding programs in zoos, if you want to exchange individuals between zoos for conservation purposes, you want to reintroduce species, you want to have a high genetic diversity.
So microsatellites are found in between genes, which are the coding section of the DNA, and they're short, repetitive strands of DNA or sections of DNA, where you tend to have 2 or 4, they're called DNA bases, repeating. So it's a very sort of repetitive pattern.
The more related species are, the more microsatellites they will have in common. So they will appear at the same spot in the genome, the locus. The microsatellite itself then can have different variations. So it is in the same spot of the genome, but it can vary in length, for example, or there could be a sequence mutation inside.
We used a pretty new technique, genome by sequencing, and it looks actually at the composition of these different microsatellites, which is a big advantage over the previous methods, because before you would just look at the length. And this can be very useful because that way there are these things called silent mutations, where the length doesn't change, but actually because you have maybe 1 or 2 of these bases in the DNA changed, it actually is slightly different. So now we can say with more accuracy, oh, this monkey is related to this monkey, because instead of just looking at the length, we also have the actual information of the DNA too.
The method that we use to amplify DNA is polymerase chain reaction. And to do so, we need primers. And primers are short DNA sequences that we design, and they are complementary to the region of the genome that we want to look at. And what the PCR does, it creates copies, in our case from the microsatellite.
So we tested a whole bunch of microsatellites that had previously been published, about 270 of them, kind of spanning decades of monkey research. And using sort of genetic databases online, we tested all of these, selected the ones that we thought would work the best.
So after screening all these loci, we selected the 45 most promising loci for further development. And for those 45 loci, we developed new primers. And then we tested those primers with high quality DNA samples. So we took blood samples from 10 different male primates, all old world monkeys. And we tested all the different loci, whether they amplified our target region.
And this was our proof of concept, so that the markers could really work on all those different species. And our second sample set then was from field samples, so fecal samples of low quality. And those 12 samples also included 2 families. And our markers indeed can show this relationship between individuals, which is important for field studies.
So microsatellites have been used in several biological sciences. So all these different fields of research can actually benefit from this panel, because we now have a new method with new primers that is work and cost efficient in the lab, has a high accuracy and reliability in terms of results, and can on top of that also be compared between different labs.